Ready, share video, so you guys can see my tired face. All right, awesome. Kahoot. So the Kahoot will be over chapter 12. I got a little frustrated with my previous class because there are some choices in the Kahoot that'll be answers that I will go over why those answers are the wrong answer. And then people will continue to answer questions moving forward that are wrong. Anyways, just I'm not like giving you a grade based on your correctness, so it's okay. But after like 5 times of choosing that same choice that I've already told you what it is in the first 3 questions, it just got very frustrating for me. So I'm not going to harp on you too much on it, but I'm going to give you fair warning that I'll walk you through it. Just let's not stress about it yet. Okay. One other thing that I pointed out. You okay? Yeah, what's up? So we are using our computers for the exam, but I can give you a paper test. It's the same exact test. It's just on paper. So you should be fine. Yeah. Yeah, let's just do paper test. Like I said, it's the literal exact same question. So the lab test, with how it's laid out, everybody has the same exact questions. Everybody has the same choices in the same order. And so this is one of those tests where it's not a bank and it's not scrambled or anything like that. Just to be clear about that. Now, it doesn't mean I'm keeping a little bit more eye on making sure that those things aren't getting spread from section to section. I know people talk about like, oh, this was on the test, be ready for that. That's one thing, but I don't need the whole test getting out, you know. As long as there's no sign that that's actually happening, being a concern, I'm not going to make a big stink about it. But let's do this. Chapter 12. All right. Make sure that I'm not missing anything up here. I am not. This Kahoot cranking, hopefully. All right, there we go. Everybody signed in. 1598735. He was on the Zoom. The average on the lab exam, do you guys want to know what it is so far? Yes, please. 70. Not bad. Better than it has been in past years. Whatever. 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. All right, we're going to get started with the Kahoot. I know it keeps like, it'll kick you out and you can join back in again. Remember, lecture, Kahoot. It's way more important that you just get at least a couple questions in. Yeah, eventually, for sure. I have ideas, of course. If you guys don't know, tattoos are expensive. Yeah, so to put into perspective, like I've gone to some very like in-demand artists that are very like high end, and you know you book it, and then a year later you get your tattoo sort of artists. So this one was about 1,000, and this one was also about 1,000, and so that's just these, and so that's not all of them, right? Some of them might have the more like mid-level types of tattoos. That's most of what these other ones are, but yeah, yeah. So it's like I think that's about how much this one was. My little doll, it was like 350, something like that. Yeah. And I mean, I have some cheap ones too. Don't get me wrong. Yeah. So this the bats that I have here were just 50, and my husband got some too. Anyways, yeah, we got it. All right, let's do it. All right, question. These are all going to be about our antimicrobial drugs. So, right, that'll be fun. So, selective toxicity. What is this concept? What's the idea behind it? Don't worry, I'll let you know when we get to the one that made me pissy. Because you guys are for sure not going to make me pissy. Right, so this selective toxicity is the concept that you want your drug to work against what you're fighting, whether it's a microbe or mental illness or whatever it is. You want your drug to work against that as much as possible with causing the least amount of toxicity to the patient. That's selective toxicity. When you get into the actual numbers, the ratio between where it's effective and where it's harmful, that's the therapeutic index. And the difference there is called the therapeutic window. Our next question. Did it kick you out? Alrighty. Sorry, someone messaged me about the lab exam, so I'm gonna have to use that here in a second. But okay, next question. When your life is just constantly something, right? What types of organisms do antibiotics treat? Right. So antibiotics only treat bacterial infections. So antifungals are for antifungals. What did you say, Kelsey? Yeah, and so you could pick more than one on this, then there's only one answer. So you can submit it after just one. So the idea is antibiotics just treat bacteria, antifungals treat fungi, antiprotozoals treat protozoa, antivirals. So everything else has its name in it. Antibiotics is the only outlier there. Huh? Kind of, yes. Right. So we have to remember that antibiotics treat just bacterial infections. Most of us got this, so it's not bad. Alrighty, moving on. That is not, so we're going to get into the ones that were getting me frustrated. These ones, the multi-select ones. Which of the following antibiotics affect the cell wall? I'm not going to harp on you if you don't get it off right off the bat. Now the questions after this that continuously frustrated me. All right, so the antibiotics on this list that affect the cell wall will be carbapenem and penicillins. All right, so all of the drugs that affect the cell wall are these ones: penicillins, psyllins, carbapenems, cephalosporins, bacitracin, and vancomycin. We'll get into vancomycin in a little bit because it's going to come up as choices in other ones. But vancomycin affects the cell wall. All right. The other two on this list, ciprofloxacin, is the point of contention going forward. I'm just saying. So heads up. Ciprofloxacin affects DNA and RNA synthesis, and ketoconazole is an antifungal. That's all you need to know about it. You don't need to know how. You just need to know that it is an antifungal for my class. All right, moving on to the next question. Which of these antibiotics affect the ribosomes and protein production? All right. So, as stated in the previous slide, again, I'm not going to harp too much when it's been one slide, but it was continuously slide after slide after slide. So once again, for our protein blocking guys, the guys that block the ribosome. So our ribosome blocking guys are going to be a group called the aminoglycosides, and there are drugs that fall under that category. I don't need you to categorize them under there. I just need you to know that if I say it's an aminoglycoside, it affects the ribosomes. If I say that it is a mycin, other than vancomycin, if it's a mycin, then it affects the ribosomes. If it is a cyclin, it affects the ribosomes. So this is aminoglycosides, mycins, right? We'll come to vancomycin in a second. Mycins and cyclins, like tetracycline. Or doxycycline, right? So those cyclins, the mycins, and the aminoglycosides in general will affect ribosome. It will inhibit the ribosomes from functioning and therefore prevent protein synthesis. Our cell wall, psilins, cephalosporins, carbapenems, bacitracin, and vancomycin. So vancomycin, and we just had it, it was the cell wall one. It is the outlier mycin. So it's the mycins otherwise, ribosomes. Vancomycin special, cell wall. Those are outlier there. Ciprofloxacin. I said it on the other side. I'm going to say it again. This is the one where people just keep choosing it for the wrong category, and it started frustrating me. Okay, so ciprofloxacin. The floxacins fall into a category of drugs called the fluoroquinolones. Anything floxacin, levofloxacin, ciprofloxacin, if it has floxacin, it is called a fluoroquinolone. Fluoroquinolones inhibit DNA and RNA synthesis. And then ketoconazole is an antifungal. Are we ready? Okay. I have faith. I have faith. Okay, so moving on to the next question. Which of these guys affect folic acid synthesis? Folic acid synthesis. Right. Thank you. So, so we're following. So this is our sulfa drugs and trimethoprim. So folic acid synthesis is all of the sulfa. It has sulfa in its name. Folic acid synthesis. All of the sulfas plus trimethoprim. Thank you for not choosing ciprofloxacin because I'm not kidding. It was just like slide for slide. And anyway, so ciprofloxacin once again is a fluoroquinolone that inhibits DNA and RNA synthesis. And then vancomycin is the cell wall. We good? We're so good, right? All right, next group. Which of the following antibiotics affect the cell membrane? Not the wall, the membrane. Polymyxin. And then bacitracin and vancomycin both affect the cell wall. And ciprofloxacin, DNA and RNA synthesis because it's a fluoroquinolone. Excellent. You're saving me so much stress right now. All right. Great work, guys. Which of these antibiotics affects the nucleic acids? Okay, so the ones that affect the nucleic acids are the floxacins, including ciprofloxacin, levofloxacin, and those guys, okay? Metronidazole, I'm gonna talk about it in a minute. Let's just leave it there for now. All right. Which antiviral is used to treat the flu? This should be a gimme. Right, Tamiflu. Good job. So flu is caused by the influenza virus, and we treat this with the antiviral Tamiflu or Relenza. They do the same thing. They both affect the N in the H1, like HN thing. It's called neuraminidase. Did you have a question, Desiree? Yeah, I bet. And it's one of those things that you really have to take early on in the infection in order to get it under control. Because otherwise your immune system is already causing damage. And yeah, man, the flu can be really rough. And I feel like people kind of take it for, what's the word for that? Take it for granted, I guess. But like, yeah, it can be really bad. It can actually be deadly. We hear about that for some people, but some people it's just mild. It just depends on your physiology. All right, which of these are antifungal? All righty. So this one is miconazole and amphotericin B. Generally speaking, if it ends in azole, it's an antifungal. And then we also tack on amphotericin B, it's an extra. Right. So we've made it through our cell wall. Psyllins, cephalosporins, carbapenems, bacitracin, and vancomycin are our tack-ons for that one. Then we went through our protein synthesis inhibitors. That's our aminoglycosides, cyclins, and mycins, minus vancomycin, which is the wall. Then we did our folic acid synthesis, guys. Folic acid, we've got our sulfas and trimethoprim. We had our cell membrane guy that was just polymyxin. And then we had our DNA guys. Those are the floxacins, and they fall into a group called the fluoroquinolones that inhibit DNA and RNA synthesis. Then we went into our antivirals. The antivirals that we have to know for this class, actual antivirals like Tamiflu or Relenza affect neuraminidase of flu specifically. We have to know that there are drugs that attack those specific proteins for HIV, reverse transcriptase, integrase, and protease. Those are specific to HIV. And then we had the antivirals that fall into the acyclovir kind of relatives. So acyclovir and valcyclovir. Valcyclovir trade name is Valtrex. And so this is the same thing. And those guys are for DNA viruses like herpes. So those are the only antivirals we have to know. And let me tell you, most of the antivirals are for a virus. They're not usually lump group antivirals. And then we have now our antifungals. So these are the azoles and amphotericin. We have one deviant of azoles, and that was metronidazole, which we saw for a second earlier. Metronidazole is the azole that went to the big metro. It treats protozoa as well as anaerobic bacteria. It might be the last question. Might, hint, right? Okay. Which of these is used as an antiprotozoal and is also used to treat anaerobic bacteria? I might have just said it. I still won't hold it against you. You guys did really great with this. Yes. Right, so metronidazole is for protozoa and for anaerobic bacteria. Chloroquinoline is a made-up thing. There is chloroquine, and I won't do that to you on the actual exam. I'm not going to put something that sounds like a real drug. What your choices will all be real drugs, and you'll have to know which real drug is the answer, okay? That will be on the exam. But chloroquine is a drug that we use to treat malaria. So it's a specific antiprotozoal group of the quinine drugs. All right, so ivermectin and praziquantel, these guys are drugs that are used to treat helminth infections or parasitic worms. And helminths are animals, and so they're the only microbes that we have that make eggs for sexual reproduction, right? The only microbes in microbiology that make eggs, right? Just a hint for that lab exam that's coming up. Okay. All righty. Great job, everybody. All right. Okay. Yes, get out. This one really, it's being real crotchety. All right, let's move on. Let's do chapter 15. So chapter 15 is going to be our innate immunity. Chapter 16, we'll talk about acquired immunity. Chapter 17, we're going to go with things when your immune system goes wrong. When it's not doing immune system things, it's doing other things. And then chapter 18 is just diagnostics. And like most of chapter 18 is stuff that you've already learned. It's just putting it into terms of diagnostics. Alrighty, so when your body is trying to basically set up defenses against the possibility of foreign things, so whether that is like actual pathogens that are entering your body or just microbes that we can count on a day-to-day basis, we basically have to have some sort of defense mechanism to keep those microbes from filling in our empty stadium seats and whatever. You can break down your host defenses into two categories, innate and acquired. Chapter 16 is going to be acquired immunity. And acquired immunity has everything to do with this cell recognizes this very specific antigen. And so that's what it does, is only see that antigen. It doesn't say all bacteria. It says this specific part of this bacteria is bad. And if I see it, it means kill. That's what our acquired immune system is going to do for us. And it has memory associated with it. Your innate system is basically like, this is a bacteria, we kill it. It's not specific, all right? So innate immunity has to do with first and second line of defense. Your first line of defense is obvious stuff. The fact that you have keratinized skin cells on the surface of your skin that's keeping microbes from getting into your body. If you get a burn, you're more susceptible to infection. We know that because you've decreased your defenses there. We have enzymes that are in your tears and saliva. We have the acid in your stomach. All of these things are part of your first line of defense. And so these are things that might have other functions too, but that they're there also to act as a first line of defense. This includes physical barriers like your skin, like your eyelashes and your nose hairs. Things like this are actual physical barriers. This includes chemical barriers like the acid in your stomach. And in your saliva, we have an enzyme called lysozyme. Lysozyme is an enzyme that breaks down peptidoglycan. That's what it does. So that's what makes up cell walls and like all bacteria pretty much. So having that enzyme there is a chemical barrier against microbes, just it being there. And then we have our microbiome that can sit and take up the stadium seats so the bad guys can't sit down. And so all of these guys play a part in that first line of defense. The second line of defense is when your white blood cells start coming into play. And this relies very heavily on recognizing self versus non-self. So first of all, immunology is the study of basically all of this line of defenses. How we're surveilling our body for foreign versus self, how we're recognizing the foreign material and then destroying it. A lot of this relies on telling the difference between self and non-self. Anything that is recognized as non-self is foreign. So you have to train your immune cells basically from whenever you're a baby developing in the womb all the way throughout as you're becoming an adult as to what is self and what is foreign. And your microbiome has a lot to do with that training. It basically helps train your body when and where you should be reacting to things and stuff like that, and how hard, strong, or however you want to look at it. So any of these markers that we can use as identifying something as foreign, we call them antigens. We learned about that in lab. These could be molecules on the surface of cells. It could be proteins. It could be sugars. It could be anything that we could recognize as foreign. And our immune system basically will evaluate them and determine whether they think it's self or foreign. All right, so once we see something as foreign, we might want to treat it as a threat and get rid of it. Autoimmune disorders is when this stuff goes awry. So when your body isn't getting that training signal properly taken care of, we might accidentally trigger our immune cells to attack something as foreign when it's actually self. And this happens a lot when we have microbes that mimic our cells, molecules. And so that's called molecular mimicry. And so, for example, strep has a capsule. The strep throat has a capsule. In its capsule, it has hyaluronic acid. We've already talked about how hyaluronic acid is part of our extracellular matrix, right? And so when you're trying to identify strep as foreign and start an immune response against foreign, you can sometimes accidentally trigger your body to attack those molecules that resemble self. And that can be temporary, but in some cases it can be permanent and actually lead to autoimmune disorders. Autoimmune disorders exist because your body sees self as foreign. There's some aspect of your body cells that for some reason got triggered as being seen as foreign when it's not. This is where we get examples like rheumatoid arthritis and lupus and things like that, is your immune system is attacking yourself when it should have been seeing it as self instead of doing that. Then when we have the inflammatory response triggered by seeing these things as foreign, the inflammatory response can be damaging. And it's typically okay if this is a short-lived thing to have some of your cells be sacrificed to get rid of a bad guy. But whenever we start having sacrifice of self to get rid of bad guy, and that goes on for a very long time, this can cause some serious health issues, which is what we see with rheumatoid arthritis. We start seeing the bone sort of overgrowing in response to trying to heal from the inflammation. And then you have loss of function. In your innate system, we're not reacting to antigens, which are very specific associated with foreign. In your innate system, there are markers that we can see as just, hey, if I see this molecule, it just means bad guy. It doesn't mean they specific bad guy. It's just something we can see as just bad guy. Some very obvious examples we've been following since minute 1 is going to be endotoxin, which is LPS. So that's just one of the molecules on the outside membrane of gram negatives. All gram negatives have it. That's not a specific thing to E. coli only, which is a gram negative, or salmonella, which is also a gram negative. All gram negatives have endotoxin. And if your body sees endotoxin floating around somewhere, that means that there is bad guy. And so your initial innate response can see that and start triggering the innate reaction. All right, so our innate immune response is pretty much instantaneous. We don't have to find the guy that sees specifically this antigen and then multiply him out and make him make all of his products and respond to that like we're going to see in our adaptive immune response. This is something that our body can do immediately. It's a very general response. So endotoxin, another one that's a great marker for bad guy is peptidoglycan. Our cells don't have that. So generally speaking, if you see peptidoglycan outside of the gut, then that's going to be a time for those white blood cells to be like, hey, you don't belong here. It's time to start our response. And another good example would be a double-stranded RNA. Even with our tRNA, it's one strand that's folded back on itself. But if we have two strands of RNA that are making up two separate strands of RNA that are making up like a genome, for example, for a virus, which we learned about is a possible thing for viruses to have, our cells never have double-stranded DNA, or RNA, I'm so sorry. Our cells never have double-stranded RNA. So if we see double-stranded RNA, it's a sign of viral infection. We don't care what virus it is yet. We just care that there's double-stranded RNA. And that's a sign that something is wrong. And so those are PAMPs. Those are pathogen-associated molecular patterns. And so that will absolutely be on the Innate test, FYI, being able to recognize that acronym. And we have special receptors called pattern recognition receptors that recognize the PAMPs. Again, these are not specific antigens to a specific pathogen. These are general markers of bad guy stuff. And there's more than just those ones, but those are some ones that are pretty common. This brings us to getting introduced to our lymphatic system. So most of us might know that your lymphatic system is tied in with your immune system, but you might not really understand how all this is going to work together. So this is where my story of talking about how a microbe will get introduced in your body and what your body's going to do is going to come into play. Let's say you're walking around, maybe picking up some clothes around your house, cleaning up, tidying the house, and you stub your toe on the side of your bed like you always do, or whatever it is, right? You stub your toe. We do it all the time. This time you draw a little bit of blood. We've broken the skin. We've actually broken our first line of defense in this particular example. A little bit of blood leaking out because we've literally damaged our capillaries and blood's leaking out of them, right? So what happens? Well, we know it'll get red and hot and swollen. We all know that. But what is actually going on with that and why? But what happens is when you have damaged cells that have been broken from that trauma, you don't even have to break the skin. You could just like bump your knee, but have some damage underneath. If there's cells that are damaged, they're releasing chemicals saying, help, I've been damaged, we need somebody to come fix this. They release these chemicals, they're called cytokines, we'll get to them in a moment. These chemicals call for the inflammatory response. So I'm damaged, I start calling for help. This was happening in your toe when you stub it. So what happens is we get more blood flow to that area, which causes the redness and the warmth. Blood is warm. And in those extremities, you'll notice the difference. And so blood flows to that area, causing warmth, causing redness, and then the fluid flows out of your blood vessels and into the tissue. That happens because when the fluid flows out of your blood and into your tissue, it's bringing with it the signals for the inflammatory response, the nutrients and factors that are needed for your white blood cells to do their job, as well as your white blood cells themselves. So they're coming out of your actual bloodstream and into the tissues in the area. And the fluid comes with it. All right, so that's why we get swelling in those areas, right? We get pain because there's nerve damage. Why do we need pain? It lets you know that something's going wrong. Like we kind of understand why pain is there. We hate it, but we understand it. So those are the 4 signs of inflammation: pain, swelling, warmth, redness. And we have words for each of those. The pain is called dolor. The swelling is called tumor. The redness is called rubor, and the heat is called calor. Those are our Latin terms. If you speak Spanish, this is easy crap for you. Yeah, this is easy. And if you know what a calorie is, it's a measure of the heat that comes off from like burning glucose or something. That's what it means. It means the heat that's released. And so calor means heat. All right. So that's what's going to be happening. So all that fluid is coming out, and then we're bringing in our white cells, we're bringing in our signal molecules to control all of this. We're putting blood into the area to help, bring the fluid there. And all this is being signaled because you had cells that were damaged and calling out for help. So that fluid moves into your tissue. And if it gives enough fluid in there, that swelling with that fluid in that area, there's a term for that. You guys know what that is? Edema. So edema is when you have fluid coming out of your bloodstream and into the tissues. And that's what that swelling is. Tumor is that what we call big picture swelling of it. But the actual medical term for that swelling from the fluid is called edema. All right. Normal situation will be, we'll get that healing underway, and then that fluid will get reabsorbed by your body. What is really going on with that fluid reabsorbing into your body, though? What's actually happening? What's happening is that fluid that got pushed into your tissues from that injury response brought all that fluid, the cells, the signals, the nutrients, the growth factors, all of this stuff to help with this process. And now it needs to go back into the body. It does not enter back into your bloodstream right away. What happens is it enters into a different system called the lymphatic system. So it leaves from the tissue and it goes into the lymphatic system. And the whole reason for that is when you're leaving the tissue and going back into the body to rejoin the circulatory system, we can't just have you straight up go into the circulatory system. But why is that? Because your circulatory system, your blood, what's your blood doing for you largely? It's got your red blood cells that have hemoglobin in them, and they're just carrying oxygen and carbon dioxide to and from places, right? As well as all of the nutrients and the energy and everything you need for your cells to function. This is a freaking open stadium. There's no microbes to make up a microbiome in your blood. So there's all these nutrients, all these cells, an open freaking free smorgasbord for microbes. We cannot allow the microbes in there. They'll just go crazy. So to protect your bloodstream, your fluid from those injured areas does not flow right back into the blood. It goes through the lymphatic system. And the lymphatic system is a filtering system. And what it's really doing, it's like a checkpoint. So if, let's say we were at war with the whole world, okay? So that's kind of what your immune system is, right? It's your body against everything else in the world. And so we have to make sure you belong here before we let you join up with the circulatory system. We can't let you into this very controlled, gated place until we are absolutely sure you belong there. And so you will go through these checkpoints in the lymphatic system called lymph nodes. Think of the lymph nodes as the shacks where the guards are. The lymph nodes themselves aren't doing crap, but it's the area where the cells are that are going to be doing the screening. And so you have to go through and get screened by the guards at the lymph nodes. And then they'll have to check you out and be like, okay, you're good to pass here. And you've got hundreds of lymph nodes from your toe in between your heart where you're going to rejoin. And you'll have to go through all of them in order, and in your little path from the toe to the heart, you're going to have to go through multiple lymph nodes and go through multiple guards, and they're going to screen you for multiple things at each checkpoint. And once you have passed all of the checkpoints, then you will rejoin the circulatory system. And all that fluid that went into your injured area can rejoin with the fluid of your blood and start reconstituting all that. And this is happening anytime that you have any sort of injury whatsoever in your body, from bumping your elbow to like losing a whole leg, right? All of that fluid that goes to that area for your immune response will have to be filtered by your lymphatic system in order to rejoin your circulatory system. Does that help you guys understand a little bit more about what the lymphatic system is going to do? All right, so here's the caveat to this. If we are taking fluid from tissue and trying to move it back to the heart doesn't pull venous blood back to it. The fact that it pumped out blood pushes the venous blood back in, right? The pump out is what still drives the blood back in. So what is driving lymphatic fluid from tissue back to the heart? The heart is not pumping your lymphatic fluid. So why does lymphatic fluid, how does it get pushed back to your heart from your extremities? Does anybody know? Skeletal muscle movement. All right. So it is pumped back to your heart through the lymphatic system entirely from skeletal muscle movement, not from smooth muscle, not from your heart, from your skeletal muscles. So when you have surgery, or if you've ever known anybody that's had surgery, what's the first thing they have you do when you get awake and you're able to? Ambulate, move around. And if you're not able to, what do? They put those little squeezy things on your legs. Yeah. Why is that? To pump your lymph. They want your lymph to get circulated and filtered back through so we can start fighting off any microbes that might have gotten introduced from being exposed to the environment. I hope this is starting to bring some things full circle for you guys. Okay, so we got to get that lymph back in. If you don't get up and ambulate, you'll just have swelling, and that swelling will just stay in that area. And so will all the microbes in that area. And so will everything else. We can't get the healing process to really start working and clearing out everything with your lymph until you start moving around. That's why when you're sick, it really is actually pretty important that you get up and move around every once in a while to get that lymphatic system moving. All right. Good. All right. That's our little intro story. Yeah. And it's like impossible. If it's impossible, it's impossible, right? I would definitely still recommend if you are sick and even if you can't walk to the bathroom or something like that, at least in your bed, try to move a little bit. Get some movement going every now and then. Yeah, it's hard, but do it, right? So this is such an issue that whenever you are completely immobile, your body can't even deal with, you get very little friction on your skin just from laying there. And your body can't even deal with just microbes being introduced that way. And you get bed sores. Getting mobile will help with preventing all that. So that's why you have to move the patients. It's to prevent the pressure points, but it's also to keep their lymph moving. So you'll come in, you might work their legs a little bit and things like that to help with that lymph flow. So what is lymph itself? So lymph is all of the fluid part of your blood, basically. And so it is plasma-like liquid that we carry in the lymphatic system. So all that fluid that comes out of your injured area and rejoins your lymphatic system with all your white cells, maybe even microbes and things like that that may have gotten in from the injury site, will get carried in your lymph. And it will get all filtered and screened before it rejoins your circulatory system. It runs pretty congruously. It sounds weird coming out of my mouth, but it runs alongside of your circulatory system. And they're separate vessels, but they do have a similar pattern in how they're going to operate. It only moves from the extremities to the heart. Once you reach the heart, you rejoin all that fluid back with the actual circulatory system. All right, and it only moves by the skeletal muscles. Here's a map of how it's laid out, all those little vessels, and we have little aggregations of lymph nodes. Now, before we move on, does anybody know how big a lymph node is? How much would you guess? I mean, nobody sounds stupid, I promise. How big do you think your lymph nodes are, roughly? Like a lot of people guess about walnut size. Does it sound about right what your guess would be? So a lymph node is about half the size of a pea whenever it's not reacting to something. It's about half of the size of a pea. So then you're sitting here like, what are you talking about? So your tonsils are these right here. Your tonsils are not a lymph node. They are lymphatic associated tissue, but they are not lymph nodes. But your lymph nodes can get pretty big, and they can be pretty big pretty often. But you're exposed to so many FN microbes all the time in your respiratory pathway, through your nose, through your mouth, through what you eat, through what you breathe, that those lymph nodes in your neck are almost constantly reacting to things. And so they will be a little bit bigger. But when they are not reacting to an infection, they're about half the size of a pea. Yes, so your biggest congregation of your lymph nodes are going to be in your neck, your armpits, and your groin. So when we saw like the Black Plague, bubonic plague, a bubo is an infected lymph node. So microbes got to the lymph node, they got to the shack where the security guys were, and they just took over the shack. And they just decided to make that their home. They're breathing there and just blowing up the place. That's a bubo. And so buboes, we would often see them come out the neck, the armpit, and the groin. These black infected buboes. And so that's what a bubo is, if you ever wondered, but it's an infected lymph node. All right, so I worked in the transplant lab at Integris. And so we would get, if somebody was brain dead, you can have your own opinions on the morality of organ donation. I'm not going to argue with you on it. But in the organ donation process, once you are declared brain dead, if your organs are viable, then what they will do is harvest your organs in a certain order and harvest your lymph nodes. They will send the lymph nodes and the organ to the hospital where the surgery is going to be taking place. We came to Integris, and we would get called in on our on-call phone at freaking 3 o'clock in the morning. I understand it, but it was still, you know, you're living your life too. So you get called in, you get to go to the hospital and take this dead person's lymph nodes, and you get like 3. You don't get very many. You have to flush the cells out of them and then put them in a little reaction with the patient's cells that you get from their blood tubes. And then you see if they like attack each other. And that's what you do. So you look to see if there's going to be any attacking going on between the patient and then this donor's lymph node cells. And if there's a reaction, then obviously that person can't have that kidney or lung or whatever it is. So we do a lot of testing before that to try to make sure that's not going to happen, from blood tests and testing the patient to see what antibodies they have. Do they have antibodies that are for markers that are likely to be a problem, and all that stuff. So we can make them match as best as we can with their DNA and everything. But then when it comes down to it, we have to actually test the match. And that happens while that kidney is sitting on ice, while that patient is in their gown, sitting in their bed in pre-op, waiting to go. All of that testing happens while they're waiting. And so they are literally waiting on you to call them and be like, we didn't have a reaction, you are a go for this kidney. And if it had a reaction, that patient gets sent home. And it happens. Not very often, because we try to do our job beforehand, but it does happen. So anyways, that's the point. So our lymphoid organs, obviously our vessels are part of it, our lymph nodes, they're just the shack where the guards are going to be working. We're going to talk about the guards, our white blood cells, in a little bit. But that's the lymph nodes. And the spleen is very similar to the lymph nodes, but it works a little bit differently. So your spleen is an organ that filters out like red blood cells when they get too old or whenever they're dead or damaged. Your spleen filters your red blood cells out. So it's a different kind of a function. But it also has a function while it's filtering your red blood cells from your bloodstream. It filters your actual bloodstream, not your lymph, your bloodstream for microbes. And so it works very similarly to the guard shack for your lymph nodes, but it's a guard shack for white cells as well, but they're going to filter your blood, not your lymph. So just in case it made it as far as your blood, or if you got an introduction of microbes that just went straight into the bloodstream, then you can filter it at your spleen. That's what the spleen does for you. All right, and then we have, I'm going to come to the primary ones in just a second. I didn't forget them. The salt, malt, and galt. So ALT, the alt of these guys, is associated lymphoid tissue. So it has similar function to lymph nodes in the spleen for filtering out guys, but they're not in a defined organ. They're not a lymph node or a spleen. It's just tissue that has a similar behavior. This is skin-associated lymphoid tissue, gut-associated lymphoid tissue, and mucosa-associated lymphoid tissue. So these are just areas of tissue that have a similar function to the lymph nodes and stuff. All right, so what about our primary lymphatic organs? So this is where like our white cells are derived from. That's why they're called primary. So we have the action of your immune system in the secondary guys. But where did your immune system come from is your primary guys. And so this is your red bone marrow and your thymus. Most of us don't know what a thymus is. Probably never heard of it. It's an organ in your body, by the way. It's about right here in your chest. It's butterfly shaped. And from birth, it starts shrinking because a lot of the training of the cells that are in the thymus happens when you're young. And then you're done training to a certain point. When you get into adulthood, you stop training as much. This can almost completely disappear. So in older folks, we just don't really see the thymus, but you would see it in a little kid. Did you know little kids don't have a kneecap? I learned that the other day. So they have like a vestigial sort of cartilage-based kneecap. And then as they get older, they actually form a patella, a bone. But they don't have kneecaps. That was wild to me when I learned that. I'm 41. And today I learned that little kids don't have kneecaps, y'all, because they got to crawl around. Yeah, it's just, and for birth process and everything like that, it's just more efficient for them not to have a kneecap and then develop one. That is wild, right? Anyways, moving on. The bone marrow. So the thymus is something that goes away when you get older because you don't need to train those cells that are there anymore. The thymus is where your T cells come from, your T lymphocytes. We're going to get into those in chapter 16. Don't worry too much about their function right now. The red bone marrow, though, this is like what's inside of your bones. It's where all of your blood cells come from, not just the white ones for the immune system, but your red ones and your platelets and stuff come from here too. And so the big major stem cell from which all of these come from is called the hematopoietic stem cell. So the hematopoietic, and this whole process of turning those stem cells into the different kinds of blood cells is called hematopoiesis. Probably heard of it. But it's this whole process of you take this stem cell, and when you send it certain signals, it'll turn into a red cell. If you send it different signals, it'll turn into one of your white cells and all their different functions that they can have. So all of those blood cells come from your bone marrow. And this is why if you have like leukemia, if you have cancer in your white blood cells, we can give you radiation to literally kill every single white blood cell in your entire body. And then take somebody else's red bone marrow. It's got all these stem cells that aren't trained yet. They can be anything and do anything in the blood. And take somebody else's bone marrow and put it into that person who we just cleared out. And now they can train those cells to be their own white blood cells. We can train them for what is self and non-self all over again from scratch. So that's the benefit of bone marrow transplants. And they're not as picky about self and non-self. So you don't have to have as strict of a match in a bone marrow donation as you do in an organ donation. There's a little bit associated there from the genetic side of it, but we don't have to worry about that yet. We're going to talk about that in chapter 16. Get ready. So all these stuff comes from the red bone marrow. All righty, moving on. We talked about all this stuff. That's why I'm kind of clicking through this. Lymph nodes, they're bean shaped. They're about the size of half of a pea. Yes. And we know where the aggregates are. Most of us are familiar with that. And you get enlargement of your lymph node when you start having a response to something. So here's a wild thing. For, let's say, COVID spike protein, you only have one B cell, one cell altogether. I don't mean one group, I mean one cell that recognizes COVID spike protein. One. So how is that one going to react and create this whole massive immune response? Well, what happens is it's a guard in one of those guard shacks. Whenever it sees that bad thing, let's say it's COVID, and COVID comes through being like, yeah, I got clearance, I get to go on to the rest of the body. That guard says, I know you. I've been waiting my whole life just for you. And what it does is it multiplies itself into thousands, millions. And then those copies start cranking out antibodies. Antibody has its different functions that we're going to get to in the next chapter. Because B cells are a lymphocyte, it's part of the adaptive immune response, but you only have one. So that's not going to do you a lot of good if you have to wait for it to multiply and then start working on making the antibody. It takes it a while. It can take it a week or two to start getting all that activated. So what do you do in the meantime? That's what your innate system is for. So it can be basic, non-discriminate, sort of basic reaction in these areas of the body. All right, so your blood. If I take a tube of your blood, we have the different kinds of blood tubes that you can draw with, right? We know we have the red top tubes, and then you have all the other colors. The reason I'm separating them like this is red top tubes are just a tube. They don't have anything in them. They might have wax in the bottom at most. That's your tiger top tubes. The ones that are red and black on the top. But the red top and tiger top tubes they don't have any other additives in them. So you put blood in there. What does blood do if you just let it sit by itself with no additives? It coagulates. So we'll put it in that tube, we'll let it clot, then we'll spin it in a centrifuge to separate the solids from the liquids. Yeah, so liquid that you get from clotted blood is called serum. If you put something in the tube to prevent the clotting, then you spin it to separate it out into its parts. The liquid that you get from that is called plasma. That's the difference. Yeah, so the liquid portion, if you add anticoagulant, if you prevent the clotting, the liquid portion is called plasma. If you let it clot and then get the liquid, it's called serum. That's the difference. If you donate blood and it's going to be transfused into another person, can you let it clot? No, there's no reversing that. But when you donate blood, it goes into a bag that has anticoagulant in it. FYI. Otherwise, it would literally just, the whole bag would just solidify in like 30 minutes. All right, so let's get into our white blood cells. White blood cells are also known as leukocytes. This is a term we need to be familiar with. Leukocytes. Leuko. This is your white blood cells. You can divide your leukocytes into two groups, granulocytes and agranulocytes. And what does granulo sound like? Granule, right? What's a granule? Like little grains? It's what it sounds like, right? So those cells might look something like this. You might have some blobs that are big linked together, sort of, maybe, big old blobs. But then you have these little, like, it looks grainy. If you stain these guys and look at them on a microscope, they have these little grains in there. That grainy look means that you are a granulocyte. That's literally how they are defined. If you stain them and they have this grainy look to them, they're granulocytes. And as it turns out, these grains that are in here are all related to the innate response. They're like histamines and other signaling molecules that are going to play a part in your inflammatory response. So those are your granulocytes. Your other group are called your agranulocytes, without granules. No grains. It's not grainy. So they'll look like this in the microscope. They'll have big old fat nucleus that'll be nice and strong, dark color. And then everything around it will look nice and smooth. There'll be no graininess to it. This is an agranulocyte. And that's literally how they are defined. As it turns out, we can break these apart further into the cells as base their functions and base their functions on that. So our two groups, let's start with the granulocytes, the ones that are grainy. And in our pictures that are drawn here from McGraw-Hill, you can see that these are grainy looking. Look at all those little dots inside of these guys, grainy. But then look at all these other guys that are on the agranulocytes side. Do they look grainy? Yeah, and that's what they look like when you stain them. So our granulocytes includes two major groups, the fills, and then another called the mast cells. For our class, these are what you need to know, okay? We're going to talk about what the mast cells do in chapter 17. I'm not going to get into it now. We are going to talk a little bit about the fills, though. Neutrophils, basophils, eosinophils. Your neutrophils are your most numerous white blood cell in your whole blood. 55% of your white blood cells are neutrophils. And your neutrophils are incredibly important for that initial innate immune response. They go up and very generally say, oh, this is a PAMP, this is LPS, this is double-stranded RNA, whatever. It recognizes this very general non-self marker and says, I eat you and I digest you. And that's what it does. It eats it up, phagocytosis. It'll go through phagocytosis on the bad guy, eat it up and digest it, and just poop out its parts. And that's all it does. So when you initially stubbed your toe and broke your skin open, we brought all that fluid in there, all those signaling molecules, and the first cells to get there are going to be your neutrophils. And they're just going to start eating up any of the damaged cells, the debris, and any pathogens in those areas. They'll just start eating them up, digesting them, and pooping out what's left over. And that's all they do. All right. The neutrophils, they'll go to the site and they'll just start eating up debris, dead cells, and pathogens, digesting it, and pooping out the waste. break it down into parts and just crap out waste. Just leaves it in the area. We're gonna come back to that though. All right, your basophils and eosinophils can have similar functions. They're usually more involved in the signaling side of things. They don't do as much phagocytosis. Your basophils are usually more associated with our inflammatory events in general. And your eosinophils are very closely associated with parasitic infections. So that means your protozoa and your helminths. So I don't know if you've noticed, but in the United States, in developed countries, we don't have a whole lot of parasitic infections. So your body doesn't know what to do with its eosinophils and your other parts of your immune system that are designed to fight off parasites. We have this built in our bodies just naturally, but that part of that system isn't getting trained as you're growing up to deal with helminths and protozoa because we just don't see them. So instead, it doesn't get regulated properly. We think that this has a huge deal to do with allergies and why we have them. Spoiler alert, third world countries where you have parasitic infections, they don't have allergies. They don't have them because their immune system is properly trained on what to do with their eosinophils and their parasitic response. Interesting? That's pretty interesting. We'll get into that in chapter 17. So your mast cells are involved in that. Those are your granulocytes. Moving on to our agranulocytes. This is two major groups. Your lymphocytes and your monocytes. For my course, you need to know two lymphocytes. I know there's more than just those two. I don't care about literally any of the others. The T cells and the B cells. Your B cells are your bombers. They might sit there and scream at your lymph node. And they might say, I see you, COVID, and I've been waiting for you my whole life. Hold my beer. And then what they'll do is multiply. And what they're going to do after that is drop bombs. The bombs that your B cells drop are your antibodies. The antibodies are what's doing the work, not the bomber, right? The bomber isn't actively going after the bad guy. The bombs are. And so that's your antibodies. Your T cells are going to go out to the area and engage and regulate everything. So your T cells are like your active tactical troops. They're very specialized specialists. They're not going to generally see bad guy, but they will see a very specific antigen and seek out the antigen. They are going to be able to go and find out cells that are infected and kill your cells that are infected. So if you're sitting there waving a flag saying, help me, I'm infected, your T cells can go out there and destroy those cells that are infected so they don't spread it to other cells near them or just kill them. It's almost instantaneous. It's crazy. We also have T cells that are going to act as those tactical troops that are going to be involved in like telling everybody else what to do. They're sending out the signals that regulate all of this. So you're not overreacting and you're not going out and just killing cells that are infected that aren't really infected. You don't want that to get out of control either. All right, that's what your T cells do. And since your T cells are going to regulate pretty much everything else, once your T cells, if you have any issue with your T cells, if you deplete your T cells, maybe you have HIV. HIV infects T cells. So HIV can deplete your T cell number, and now you can't regulate your immune system. And so now you're suddenly incredibly susceptible to infections. Again, we'll get into that in chapter 17. We'll introduce T cells and how they work in chapter 16 and talk about problems with them in chapter 17. So those are your lymphocytes. Those are your very specialized adaptive immune cells, your T cells and B cells, and they give you memory. Once they have seen a thing, they remember, and they stay around to fight the good fight when you see it again later. Then we have our monocytes. Monocytes are big old white blood cells that have a very general action, sort of like your neutrophils do, but they're an in-between guy, and they flow around in your bloodstream. Monocytes are found in your bloodstream. Once they start getting signal from your body, let's say you sent out signals for help from your toe, they're going to follow those chemical signals to your toe. Once they move out of the bloodstream and into the tissue, they'll differentiate into that, and they'll be devoted to that area, and they'll always come back to that area. So when they come out of the bloodstream, so monocytes are in your blood, Once they move into the tissues, they will turn into either macrophages or dendritic cells. These guys are very similar to each other. They look different, but they have very similar functions to one another. And it just depends on what part of the body they're in. I'm never going to have to make you know really which part of the body, other than dendritic cells are very commonly associated with the skin. So odds are, if you bumped your toe, your monocyte's going to move out of your bloodstream and turn into a dendritic cell, because that's near your skin. Just saying. All right, so our macrophages and dendritic cells, these are our heavy lifters in the battle. Why? What they do is, your neutrophils, they're just non-discriminately going around. Are you self? No? Okay, I'll eat you up. I've been told to come here and eat up non-self. I'm just going to eat up non-self and just crap out what I digested. And they're just leaving it there. The macrophages are there to come and clean up the mess. So after your initial neutrophil attack, your macrophages are coming in there sorting through all the rubble and saying, okay, let's pick up all these parts, and I'm going to go and show them around to the guards at the guard shacks and see if they recognize any of these parts. And that's where that comes into play, where your B cell that was sitting there just counting the hours, waiting for years to see COVID spike protein. So one of these macrophages picked up a COVID spike protein from your injury site, your infection site, or a bacteria from where you stubbed your toe. It brings up a piece of that guy, shows it at the guard shack, and say, anybody recognize this? And that guy, 10 years later, you better bet. I've been waiting for this moment. And they get to go to work. All right, so that's what your macrophages and dendritic cells do. They're to clean up the mess of the neutrophils and go and start activating your adaptive response. All right, that's the big story with that. So you can see how this is going to be quite a story. Moving on. Those white blood cells, they don't just float around randomly in the tissue. They are traveling along a network called the mononuclear phagocyte system. It's just a network of fibers and tissue on which these white blood cells can travel between the tissues, between the organs, between the cells and everything. So this is how they're getting around. They're crawling along the highways of your MPS. We talked about our monocytes. They're in your bloodstream, and once they go into your tissues, they can become differentiated. Some examples of very specific differentiated, like macrophages and dendritic cells. The Kupffer cells in your liver, they're macrophages that basically just live in your liver and wait out for bad things. All right, those are your Kupffer cells. The alveolar macrophages, they say what they are. They're macrophages in the alveoli in your lungs. They just stay there and wait for bad guys in that area. The Langerhans cells in your skin, those are dendritic cells in your skin. And then in your brain, you have these microglia. These are special macrophage type cells that don't have as strong of an immune reaction. So all of the guys that are in the rest of your body, we can cause damage. Your tissues will recover. You'll be fine. But you cause damage in your nervous tissue, I'm pretty sure we are all aware of the fact that your nervous system can't recover from that. You cause damage in your nervous system, it's permanent. There's no healing that. So we can't have massive inflammatory response in your nervous system. It's a privileged area. And so these guys don't react as strongly to things. So it's special for these tissues and how we need to react to certain microbes there. The signals that we send out asking for help or to control this whole thing, the signals for the cells are called cytokines. I need to know that one. There's 4 major groups of cytokines. Pro-inflammatory, like it sounds, it encourages all of your inflammatory response. Sends out signals saying, yes, more inflammatory, please bring more guys in, start making histamine, all these things. You're anti-inflammatory. These are signals that are going to start calming that response down. If you're done fighting and there's no microbes there, we can start sending out signals saying, okay, thanks. We're done, we're good now. Your vasodilators and vasoconstrictors. If you stubbed your toe and you see blood there, you've opened up your bloodstream to microbes, to the environment, right? Your body doesn't want to let that blood rejoin into your body because it might have microbes in there. So we can vasoconstrict to prevent your veins from running as much blood back to your heart. And we can vasodilate your arteries that are bringing blood to the area. And so we can control blood flow to the injured area and from the injured area through our vasodilators and vasoconstrictors. Then we have our growth factor cytokines. These are signals that are going to encourage your adaptive immune response. Oh, he doesn't know I'm at work. All right. These are signals that are going to encourage your T cells and B cells to start multiplying and start doing their thing. So when we find the soldier that knows what COVID is, encourage him to proliferate as fast as possible. We want to send signals out to encourage that. And if you don't get signals that encourage that, you might be like, I'm pretty sure I know you, you're COVID. But it could just be some guy that looked like COVID. We don't want to encourage you to start reacting to something we don't need you to react to. Because the bombs and the tactical soldiers that are coming out to kill your own cells, we don't want to start that fight if we don't need to be starting that fight, because it'll cause some serious damage. That's all going to be regulated by all these signals that are involved in the immune response. Any of the signals guiding any of this stuff is called cytokines. An example of a cytokine is histamine. And goodness, you guys know what that is, right? We know that it's going to be leading to the inflammation that's associated with that. We definitely know there's vasodilators there, getting your red eyes, the itchy eyes, and all this stuff associated with inflammation. Inflammation has its purpose and does a role in fighting off microbes that hopefully we'll get to today. We might not get to it until next week. That's okay. We kind of touched on our physical barriers, so I'm not going to go over them too much. But the waterproofing protein in your skin, the way I say keratinized skin cell layer, keratin is a waterproofing protein that helps with creating that layer of your outer skin cells that makes it impenetrable to microbes. So keratin is a waterproofing protein. Our hair follicles and our glands in our skin, there's oils and things there that can be antimicrobial. It can help with flushing these guys out. The saltiness can be inhospitable to a lot of guys. The moistness of your mucous membranes, we can have flushing of these tissues by producing mucus in this area that constantly is moving these microbes out. It traps the microbes so they can't get to the tissues and all of this stuff. We have nasal hairs that are literally trapping particles from getting into your nose, or your eyelashes protecting your eyes from getting contaminated. We have the cilia in your respiratory tree constantly moving mucus out to get rid of microbes that may have gotten down in there. In your GI tract, we have the trickle of urine flushing out your ureters and your actual urethra. We have vaginal secretions that are helping out with flushing of microbes into that orifice. And then we have microbial antagonism, that your biome is taking up the stadium seats, using up the nutrients, and maybe even producing antimicrobial things that can be inhospitable to pathogens in the area. Lysozyme is an enzyme that your cells can make that breaks down peptidoglycan. We see this in your tears and in your saliva. This is a chemical defense. You have lactic acid, especially like if we're talking about lactic acid in your sweat, electrolytes in your sweat. That acidic environment of like the vagina and the reproductive tract that is inhospitable to some microbes. Your stomach acid, your bile. Bile is actually alkaline, and it also just a digestive juice. It'll break down the microbes just in general. So we have that. Semen has antimicrobial chemicals in it. Okay, so moving into phagocytosis. So we've been talking about some phagocytosis up in here. Let's get into the nitty gritty of it. Phagocytosis is when you have white blood cells eating up foreign guys, bad guys. When the cell comes up to the thing, they'll come and they'll engage with it. And they're seeing if you look foreign or self. If we see that you look foreign, it could be you have a PAMP, or it could be you're not sending self signals. We interact with you and we decide you are not self. We'll eat you up and break you down. We'll break you down into your parts, and this is the process of phagocytosis. The main phagocytes in our body are neutrophils and then monocytes and all their derivatives. This includes macrophages. Those are the main ones. There's other ones, but definitely know that neutrophils, monocytes, and then macrophages are a differentiated monocyte, okay? It's just a subset of that. All right, so let's talk about these guys a little bit. Neutrophils, the most common of your white blood cells, they are the major component of pus. So when you have an infection and you see that there's pus associated, whether it's from a zit on your face or because you got a cut that got a little infected, pus is mostly neutrophils. It's some bacteria and it's some fluid and things and clotting factors and things like that, but it's mostly neutrophils that went in there to go eat up and digest and kill everything in that area. And if you have a higher than normal neutrophil count, it's almost assuredly a sign of infection that's somewhere in the body. So I test your blood and I do your CBC count. When we look at your neutrophil count in that, in your white blood cell counts, your neutrophil count is high, I'm going to assume you have an infection somewhere in your body. And I just need to figure out where it is and everything like that. All right. So these are your primary phagocytes, and they're going to be your like earliest responders in an infection. Anytime you have damage in the body, these are your early responders. Eosinophils are mostly associated with parasitic infections. They have a little bit of a phagocyte role, but it's not that important. Just definitely know that eosinophils are associated with parasitic infections, protozoa and helminths. All right, now let's move on to our monocytes and our macrophages. Again, monocytes are in your bloodstream, and they transform into macrophages or dendritic cells when they move into the tissues. All of our phagocytes have these structures inside of them called lysosomes. They're little vesicles filled with digestive toxic substances. And basically you get a chemical signal from your chemokines or your cytokines, same thing, cytokines, cell signals. You get your cell signals in, stub your toe, cells start screaming for help. We start seeing that cry, then we'll start moving towards where that cry came from. That's chemotaxis. We're responding to the chemical signals. And we'll eat you up by ingesting you. Once we know you're not self, we'll eat you up. That's ingestion. And then that vesicle that we ate you up into is called a phagosome. A zome is just a vesicle. A phagosome is the vesicle that you got ate into. And we'll take that lysosome, the vesicle with the toxins in it, merge it with the phagosome, and start digesting you. That new vesicle that's been merged is called the phagolysosome. It takes about 30 minutes for your cell to digest bacteria that ingest it. Then that leads to destruction, and we will expel the waste. I think that's where we're going to end it. This is all going to be basically governed by interacting with PAMPs and making sure you are non-self. You get those signals from those cytokines, and all of this will be basically controlled by all of these things happening that way that they're supposed to. We will pick up with inflammation. We've already kind of talked about these points, the redness, the warmth, the swelling, and the pain, and what those words are for them. We'll pick up here and get in more detail on it next time. Good? Any questions on any of this stuff so far? I hope it's starting to sound a little cool to you guys. Good question. No. So we'll have chapter 15 due for the quiz for that. Chapter 15 quiz will be due whenever we finish chapter 15. So if we finish chapter 15, which we almost assuredly will next week, it'll be due next week. And we'll continue with that trend. If we keep having to push them back, I'll update it. That's a really good question. Let me make note of that before I forget. You absolutely need your lab coats. You get points off if you don't have it. All right. Any other questions? So we're going to head up to the lab. Well, I'm going to let you into the lab. Go to your normal spot. All right. And once you get to your spot, please pull up your lockdown browsers. Like get set up for the password immediately. So how are we going to go around like this? We'll talk about that when we get there. Yeah. Was it run around like monkeys and turn off? God. There will be a timer and we'll rotate with the timer. It's a little tedious, but it just will make sure everybody's seen everything. And it's just like we're going to the microscope, figure out what it is. Basically, yeah. It'll make sense when we get it started, I promise. Okay. One of the questions, like, where do you get it from? One of them is. Yes. It's going to be the helmets. So I'll ask you one of the helmets. How do you get this helmet? And then you'll have to choose from the choice. How it's transmitted. Where you get it, like from people or whatever. So I haven't studied that one well, so I didn't know if that was. There'll be one question. From the helmets. Yeah, from the helmets, yes. Move to chapter 15 quiz next week. I just don't want to forget this because you guys know that we'll all get sidetracked. Okay.